Here at Biomodels, our goal is to provide translational models to our clients. So there's a number of criteria that these models should fit when we're trying to model and treat a disease, these preclinical disease models. First and foremost, we want the model to replicate the clinical condition. And we'd like to see that the underlying biology should be the same or very similar to the human disease or the pathology that's seen in human disease. And ideally, these models are cost-effective and can provide actionable information so that we can assess the treatments in an efficient way. Finally, we like to see that the endpoints should be translatable to those used in clinical trials. So there's a number of clinical endpoints that are often assessed in pulmonary disease, and these are pretty common to the diseases that I had previously mentioned. One being biomarkers, and these are assessed in both sputum and serum, as well as in tissue. So one thing that will often be looked at is the type of the inflammatory cell in this fluid or tissue, as well as inflammatory mediators such as cytokines. Something else that is looked at is lung function, and there's a number of different components of lung function that can be measured, including forced expiratory volume, forced vital capacity, airway resistance, lung compliance, and a number of other measures of lung function. Additionally, exercise tests are used to assess lung function as well, such as the 6-minute walk test or an endurance test. And so we have a number of ways of assessing all of these components within the disease models that we offer. So first and foremost, we're often looking at a number of different biomarkers, pulmonary disease models, one being within the bronchoalveolar lavage fluid. And so in order to gain access to this, first we will tracheotomize the lung. The lung is then washed out with PBS, collected, then spun down. And that gives us the ability to look at both total cell counts as well as differential cell counts, including macrophages, eosinophils, neutrophils, and lymphocytes. And here to the right, we can see under the heading BAL cells, in a naive normal mouse, you don't normally see too many cells. The cells that are seen are primarily macrophages, and that's normal in the normal BAL condition. If you then look at the picture under LPS, this is an acute lung injury model in which LPS was administered to the animals 2 days prior to taking the bronchoalveolar lavage fluid. As you can see here, you see a lot more cells, as well as the majority of the cell type here is neutrophil. And so this is what defines the inflammatory components of this disease model. In addition to looking at the cells within the bronchoalveolar lavage fluid, we can look at different inflammatory mediators or total protein in the supername of that component. And those are also very indicative of disease, and we'll show some more specific data related to that in the specific disease models. So in addition to the BAL fluid, we often look at circulating cytokines within serum or cytokines or other markers within lung homogenate. And that can be a wide variety of different commercially available kits to assess that. Another big assessment of disease is done through histology. And so when we collect lungs at the end of a study, the lungs will often be insufflated in formalin, embedded in paraffin, and then sectioned. And then we have the ability to stain with a number of different histological stains, one being a hematoxin-eosin stain to assess general morphology as well as inflammation. And with that stain, we can gain a lot of information about cell types that are being found within the lungs. Additionally, we can stain with Mason's trichrome, which will look at levels of fibrosis and collagen deposition. Additionally, we can use a picocereus red stain for quantifying levels of fibrosis. And then in asthma models, we'll often stain with the PAS, which will look at mucus and assess levels of goblet cell hyperplasia. And then if there's a more specific marker that's of interest, we can always perform a immunohistochemical stain to assess levels of that marker in disease or non-disease states. And so this picture here at the bottom is showing lungs from both naive and animals that were administered bleomycin in the pulmonary fibrosis model. So while in the naive animal, you see nice healthy alveoli, a lot of open space. See in the bleomycin administered lung that a lot of the lung architecture has been obliterated and overtaken by the fibrotic material. So something else that we assess in the disease model is lung function. So we have a device here known as the FlexVent, which is a mechanical ventilator. The terminal procedure that's performed at the end of these disease models. The mice are anesthetized, tracheotomized, and attached to this ventilator. And this allows for the ventilation of the mouse and forced oscillations. And using this device, we're able to generate pressure volume curves, broadband forced oscillation, and in assessing hyperreactive airways, we can nebulize methylcholine and assess airway hyperreactivity.